CRISPR is a revolutionary gene editing tool. Think of it as molecular scissors for DNA. This technology has transformed the way scientists approach genetics, making it possible to edit genes with a level of accuracy and efficiency that was unimaginable just a decade ago. It uses a guide RNA to lead the Cas9 enzyme to a specific DNA sequence where Cas9 makes a precise cut. This targeted approach allows researchers to zero in on a single gene out of billions of DNA letters, minimizing unintended changes and maximizing control. Scientists can then remove faulty genes or insert new ones, making CRISPR a precise biological machine. This opens up the possibility to not only fix genetic errors, but also to enhance certain traits or study how genes function in living organisms. Remarkably, this system comes from bacteria, where it acts as an immune defense against viruses. Bacteria use CRISPR to recognize and destroy viral invaders, a natural process that inspired scientists to adapt it for gene editing. Scientists adapted this natural process for use in human and animal cells, turning a bacterial survival mechanism into a powerful tool for medicine and research. The potential is vast. CRISPR could help correct genetic disorders, offering hope to families affected by inherited diseases that were once thought to be untreatable. It could also tackle complex diseases like cancer, by targeting and editing the genes that drive tumor growth or resistance to treatment. We're entering a new era of genomic medicine, where treatments can be tailored to an individual's unique genetic makeup, and new therapies are being developed at a rapid pace. The possibilities seem almost endless, from curing rare diseases to improving crop resilience and even combating pandemics. It's an exciting time for science, as CRISPR continues to push the boundaries of what we thought was possible in biology and medicine. Cancer isn't just one disease. It's hundreds, each with its own genetic fingerprint. Cancer cells are our own cells gone rogue, growing uncontrollably and spreading through metastasis. Traditional treatments like chemotherapy and radiotherapy can be harsh, often harming healthy cells and causing severe side effects. Cancer's ability to mutate makes it resistant to many drugs, turning treatment into a constant battle. No single therapy works for everyone, so personalized medicine is the goal. We need smarter, targeted treatments that spare healthy tissue. This is where new science like CRISPR can make a real difference. The challenge is vast, but so is the potential for breakthroughs. Smarter solutions are urgently needed. Our immune system is a powerful defense, but cancer can hide from or disable it. Immunotherapy boosts the immune system, training it to recognize and attack cancer cells. Some drugs release the brakes on immune cells, letting them attack cancer more effectively. Other therapies involve engineering a patient's own T cells in the lab to better target cancer. These supercharged cells are returned to the patient, acting as a living targeted medicine. Immunotherapy has transformed outcomes for some patients, offering hope where there was little. It's a more natural, personalized way to fight cancer. The future of treatment is harnessing our own biology. CRISPR is making immunotherapy even more powerful, transforming the way we approach cancer treatment at its very core. This revolutionary gene editing tool allows scientists to make precise changes to DNA, opening up possibilities that were unimaginable just a decade ago. By editing T cells, scientists can remove genes that act as brakes, like PD-1, making T cells more persistent cancer fighters. These engineered T cells are now able to stay active for longer periods, relentlessly seeking out and destroying cancer cells that would otherwise evade the immune system. CAR T-cell therapy is a prime example. CRISPR can precisely insert a gene that helps T cells home in on cancer. This targeted approach means that T cells can be programmed to recognize and attack specific cancer markers, making treatments more effective and reducing harm to healthy cells. This leads to safer, more potent treatments that offer hope to patients who previously had limited options. The ability to fine-tune immune cells is changing the landscape of cancer care. Traditionally, CAR T therapy is personalized and costly, but CRISPR could enable off-the-shelf T cells from healthy donors. This innovation could make advanced therapies more accessible and affordable for people around the world. Editing out certain genes prevents donor cells from attacking the patient's body, making universal treatments possible. This means a single batch of engineered cells could help many patients, speeding up treatment and saving more lives. CRISPR also enhances immune checkpoint inhibitors, making T cells permanently active against cancer. By removing the natural off switches in immune cells, scientists can ensure a stronger, longer-lasting attack on tumors. Combining these strategies could revolutionize cancer therapy, creating a future where cancer is not just treatable but potentially curable for many. The precision and flexibility of CRISPR open new doors, allowing researchers to dream bigger and move faster than ever before. We're witnessing a new dawn in cancer treatment, one where hope, innovation, and science come together to change lives. CRISPR is a groundbreaking research tool, allowing scientists to dig deeper than ever before into the genetic roots of cancer. By editing DNA with remarkable precision, researchers are uncovering why some cancers resist even the most advanced treatments, and how genetic mutations drive the disease's progression. Across the globe, clinical trials are underway, testing CRISPR-edited cells in cancer patients. Early results are encouraging, with some patients showing signs of improvement that were once thought impossible. These trials represent a critical step toward bringing gene editing from the lab to the clinic. Some patients with advanced, treatment-resistant cancers have responded well to these experimental therapies. However, scientists caution that much more research is needed to confirm both the safety and long-term effectiveness of CRISPR-based treatments before they can become widely available. The technology's precision is improving rapidly, but the risk of off-target DNA edits, unintended changes to the genome, remains a significant concern. Researchers are working tirelessly to refine the process and minimize these risks. Alongside scientific progress, ethical questions are front and center. Who will have access to these life-changing therapies? How do we decide who gets treated first and who might be left behind? Ensuring fairness and equal access is a challenge that society must address. The conversation includes patients, families, doctors, and policymakers, all working together to shape the future of cancer care. The promise of CRISPR is enormous, offering the potential for more personalized, targeted, and accessible treatments that could transform lives. If successful, CRISPR could truly revolutionize cancer care, changing the way we diagnose, treat, and even prevent the disease. We're on the cusp of a new era, where fighting cancer with our own edited genes is becoming a reality, not just a dream. The future looks brighter than ever, offering new hope and possibilities in the fight against cancer. Science is rewriting what's possible, opening